A very good evening, students. In the last decades, we have discussed the just introduction of defining cell culture, right? So, what we talk about what is cell culture, what is a very for otherwise the applications of animal cell culture, so the research as well as the laboratory, and also the pharmaceutical perform production, cell production, monocle production, so on. So, we have discussed. Today, now we're going to study, otherwise, we'll get more later. So what is animal cell culture media, right? So last class we discussed the in artificial environment we are going otherwise cultivating the human cell lines, mammalian cell lines, otherwise the insect cell lines, viral cell lines in a aseptic condition by providing the appropriate carbon dioxide and humidity and temperature. And also one more thing that is essential for the growth, that is a proliferation and proliferation of a particular cells. It requires the cell culture medium, right? So medium is required for the cell growth and survival. So we'll discuss what is a requirement for the animal cell culture media. And before choosing the any kind of the media, so how we have to select the media? We have varieties of media, right? So you know, study the media for the, for example, for bacterial growth, there will be media, will be broad, right? So otherwise the nutrient, the nutrient broad. So we are cultivating in that, we'll add just a bit of yeast extract and here, right? So like this for the, in order to grow to like the bacterial cells, otherwise in order to revive the bacterial cells, we'll use the all this like the ingredient to prepare the medium. So now in case of animal cell culture, so there is a mammalian cell culture or whatever the cells we are cultivating in the laboratory. So we have to choose the media. This is depend on how we have to choose, whether we have to choose a chemical defend media, otherwise the serum free media, serum containing medium. This all depend on to what type of the cell we are culturing, okay, and what purpose we are culturing. Okay, is there any particular reason or is a particular rule to establish the cell lines and so you have to carry out particular research and particular outcome from that cell lines. So depending on that, you have to choose the particular cell lines. And we can generalize that this cell line, this culture media we can use for all the cells, depending on their growth factor. Some of cells are very sensitive. So in that case, we have to use the particular media. Okay, and we cannot use the all the and and also it depends on the purpose of cell culture and also the availability in the laboratory and also specific growth requirement. So whether you want to express any proteins, otherwise in order to study the vaccine part, otherwise if you want to develop any recombinant vaccine from that particular cell lines, that also depends on the what type of the cell you are like culturing. And one more thing, in order to establish the cell culture, before going to the cell culture media, what is the basic requirement and the benefits of like the equipment used in the laboratory? We will discuss few like equipment generally we use in cell culture laboratory. So you know that we need the LIF that is in the form of the biosafety level. Usually the biosafety level 2 it is required for the cell culture. Okay, and microscope where these are the example you can see in this slide, right? So microscope, vacuum pump for any filtration, say any plant extract or whatever you prepared. So that you have to do like the filter. We can't use a normal like vacuum filter paper. If you use the vacuum pump, so you can use a very small board or like the size vacuum pumps or the membranes will be there so that you can filter and extract the plant extract and you can use for the other cells in order to synthesize nanoparticles and so on. And carbon dioxide incubator, right? So, no, like 5 to 10% carbon dioxide incubator we have to maintain in the animal cell culture for the cell growth and development. And the centrifuge, you know, the general cell centrifuge and also water purification, that is a double distillation, like the double distilled water we need should be there. And the cell freezing, you know, to freeze the cells, we usually generally use the liquid nitrogen, means that can it contain the minus 156 degrees Celsius, like the liquid nitrogen. So we have to store the particular cells. And also we can use them any like the colony counter. Otherwise, you know that all these other the centrifuges and ultracentrifuge and many other equipment generally we use for the conditions. And some of the quality tests for the high quality serum using the mammalian cell cultures, sterility, no growth in the varieties at the real temperature, and mycoplasma and the viruses contamination should be completely absent. Okay, and efficiency of the cell growth come in the compared cell growth in the T class. So last time we have seen some of the plants used for the cultivating the cell, right? So in that we have to cultivate. We can't use our normal chemical plants what we are using for the like growing the bacterial cell, otherwise a normal test tubes or bacterial cell revival. We can't use all those things. We have to use a particular adaptive class for the global cell culture. So, and the pH should be appropriate, usually sulfuric, sulfate, in that range, right? These are some of the care we have to take in the cell culture. And some of the contamination in the cell culture. So cell culture established is a very, very like risky task. Usually cells are when the cells are prone to contaminate. Okay. The contamination, we don't know where the contamination happens. So even though we are using high sterility and fumigation and all the outlet everything we are using also, but still the cells undergoes the contamination there, right? So the contamination either in the form of chemical contamination or biological contaminations. Sometimes the FBS and all you are preparing 5% of the FBS, otherwise 10% FBS. While preparation itself, it might be the contamination will be there, right? So that it will affect the growth of the cell. And biological contamination, we don't know biological contamination, it will start, right? So biological contamination in the form of bacteria, and these are the microorganisms, right? Fungi, mycoplasma, these are the, like even though we are like growing the cells in the aseptic condition, so sometimes this all like biological like agents, bacteria, virus, and the mycoplasma generally affect the cells. And chemical contamination may affect the animal cell culture growth in the form of the mediates when contaminated while vibrating, otherwise how you are seeing receiving subculturing during that time. So, like sometimes by mistakenly the contamination will be there from the preparation of phosphate buffer, otherwise a toxinization in order to cells we have to tissue disaggregation in order to do that, we'll do the toxinization, either in the pro-toxinization or as a long toxinization. So in that also it will affect the our cells. And incubator, sometimes you're not maintaining the appropriate like the incubator, you should even though you're maintaining the 37 degree, you should only that incubator should be for the animal cell culture. So you can see that bacteria cells also we can grow in the 37. We should not place your animal cell culture plants or whatever other bacterial culture or whatever. So that time the cross contamination is there. These were the some of the like the contamination part of the ideal processing for the animal cell culture. So major equipment we have discussed in the previous slide, right? These are the like the incubator, carbon dioxide incubator, it's maintaining around the 37 degree, 5 to 10% carbon dioxide, and biosafety level cabin. This in the form of LAF. You can see all the aseptic like maintenance we have to do whenever we are doing the animal cell culture, and how to seeding, reseeding, and this the pink color, otherwise the red color shows the media. So media commercially available media, so we can for feeding the cells, otherwise seeding the cells. Okay. So now we'll discuss the basic component of animal cell culture media. So animal cell culture media, so there are 10 basic, like the most important like ingredient in animal cell culture media components. Mainly in the form of organic salts, inorganic salts in the form of magnesium, sodium, potassium, all these are the like the nitrogen sources, otherwise in the form of acids, energy source in the form of glucose, fructose, and vitamins, fat soluble content, and nucleic acid precursors, and antibiotics, pH, hormone, everything we have to like add into while preparing the media. So nowadays the commercially available media is available. So it's depend on your cell interest. You have to just purchase the media and you can like continue your and the cell culture work. So now we'll first discuss what is the culture media. Okay, culture media is, as we discussed, it is made up of the varieties of ingredient in the form of carbon source, nitrogen sources, or its antibiotics, hormones, amino acids, nucleic acid precursors. So, right? So, culture media contains a mixture of amino acids and glucose, salt, vitamins, other nutrients. Okay, They are in the form of this, all this powder form, they are available in the form of powder, otherwise it is in the form of liquid form for the commercial supplies. Right, as I said, no, nowadays no need to prepare like adding the like this ratio salt, this ratio, this may change your salt, no. So nowadays it is commercially available. You just either the suspension media you want, otherwise a solid media if you want to just like place a powder, then you will get a mixed culture media. And buffering system, so pH regulation is very, very important when you are doing for the animal cell culture media. So one, we can maintain the animal cell culture media with their natural buffer system that is in the form of carbon dioxide and carbon dioxide and CO3, like the balancing system. So another thing that is usually, I will say, we have to maintain the carbon dioxide for the cell around 5 to 10%. Okay, the carbon dioxide incubator. If you go with a natural buffer system, this is a low cost and toxic. So one more, if suppose this like buffer system is not working, so in that way we can go with a chemical based, otherwise AP is okay with a chemical based buffer system. This also maintains the pH around 7.2%. 4. Okay, so they are control the gas like not required for the 5 to 10% control mechanism here. So this itself act as a control mechanism. So another thing is they are very expensive and high concentration of this buffer in those cells will give that toxic. Okay. So as we said that carbohydrates are a major sugar component of our like we need by the whatever the sugar content in the form of glucose to glucose. So, and the media contains the glucose, glucose, and sometimes the fructose, glucose is the disaccharide. And protein and peptides, you know that is a protein and peptides is a product of the culture media. So amino acids in the form of albumin, transfer, and fibrinogens, they are also very important during the cell media. Okay, and one more thing, important thing for the any kind of a serum media. Serum is very much important. Serum is a rich of protein that includes the salt, the transferrin, and filtering, and the fibronectins. This serum is very much important for the cell growth, regulation, development, proliferation, and all of those. So they act as a very effective because it is a major essential part of the immunity. Some of the cells they will be happy, means they will be doing only in serum. Yeah. And sometimes serum itself attacks a toxic if you go for the high concentration of the cell. Some of the cells they are very sensitive for the serum. Right? So in that time we have to go with the serum. And next we have four different type of technical students. One is natural media, artificial media. So under the artificial media, we have let's say the free media, serum containing media, and defined media, and like protein and free media. These are the four artificial type of the cell culture. We are a little bit familiar with the subject media. So in this class, we'll discuss the four different types of the culture media. We should keep in the mind that a media is required for the growth and development of any kind of cell lines. Not only the human cell lines, we could take cell lines, viral cell lines, mammalian cell lines. In order to establish the animal cell culture, we need the medium, right? So the media composition is made up of carbon source, nitrogen sources, and also antibiotics. Antibiotics inhibit the growth of contamination, contamination of the bacteria, mycoplasma, fungal. Not provided, we add the antibiotics also. And buffer system, we discuss the natural buffer system or the secondary buffer system, so and so. And as we discussed, serum is essential in the media, in the media. So serum acts as a precursor for fermentation, the growth of fermentation in the particular cells. So this includes it has a rich of the albumin transfer. These are very much important in the cell growth and development, right? And the serum sometimes if you go for the high concentration, and sometimes the some of the cells are very sensitive to the serum. They'll don't want to serum. So in that time you have to avoid the serum. Right? So next class we'll discuss the four different type of the serum like the media used in the picture. Right?